Thank you. So to introduce our speaker for this evening, I'd like to welcome Jonathan Romiguier, whose research focuses on the links between species ecology and molecular evolution. So in particular, he studies genome evolution in species with eusocial lifestyles or atypical reproductive systems. And his research focuses on Messor harvester ants, apologies. And now I'm just going to hand over to Jonathan to explain everything about these wonderful species. Thank you very much.
Thank you, Zoe, for this introduction. So I will present you here my work called One Mother for Two Species, the Complex Life Cycle of Harvester Ants. And so to start, one of the most basic assumptions in biology is that living organisms give birth to same species individuals as part of their life cycle, okay? That's kind of obvious. Cats give birth to kittens, humans give birth to baby humans, and chimps give birth to baby chimps. So I gave some example of vertebrates, but obviously it's also the case in clades that interest us today, which are insects. And it's particularly obvious that living organisms give birth to same species individuals in asexual species, because the offsprings are morphological and genetic copies of their mother, such as in these stick insects. But so this is obvious that they belong to the same species. It can be a little bit less obvious in sexual species with a strong sexual dimorphism, because the morphology between a male and a female can be so different that they may appear as different species. But even in this case, obviously, they share the same genome and they belong, obviously, to the same species. And can be a little bit less obvious also in eusocial species, so social insects, such as these ants, because ants can produce at least 3 different phenotypes, 3 different morphs. So the queens, the males, and the workers that is a kind of helper caste that help the sister queens to pass on the DNA to the next generation. And I actually told you 3 morphs, but actually it can be way more than 3 morphs, because in some ant species or termite species, you can have a large diversity of worker caste. But whatever the diversity of phenotypes produced by a female, again, they all belong to the same species, because from a single genome, by phenotypic plasticity, the single genome can produce a various diversity of morphs. But as you know, in biology, there are few rules, but many exceptions. And there is now an exception to this norm, to the fact that living organisms produce same species individuals. And this exception to this norm is in Messor, where there are some queens that need to produce offspring that genetically and morphologically belong to different species. And to explain you that step by step, I will do that in 3 parts. So first, with the first part about generality, about eusociality, where I will explain you how from a single genome we can go to different castes via phenotypic plasticity. Then in the second part, we will focus on a specific genus of ants, the Messor genus, where we will see how we can go from phenotypic plasticity to genetic caste determination. And then in the third part, we'll focus on this species, Messor, where we will see how we can go from this genetic caste determination to cross-species cloning and having queens that give birth to different species. So part 1, Eusociality. So from single genome to this large diversity of castes via phenotypic plasticity. So one of the main tour de force of the eusocial genome is that a single genome can give birth to 2 extreme phenotypes. They can give birth to queens that are extremely long-lived. Ant queens can live for up to 30 years, which is very high for insects of this size. And they are also hyperfertile. They can lay some several millions of eggs per month. And on the other hand, you can have workers that are all the reverse. They're short-lived. They can live for up to 2 or 3 years, and they are mostly sterile, okay? And so from an evolutionary point of view, social insects are very interesting, because an ant is kind of like of a Darwinian demon. That is to say that usually animal species, you've got a kind of trade-off between fecundity and longevity, meaning that species that have a large fertility tend to be short-lived. But in the case of ant queens, this trade-off seems to not exist because they both very fertile and long-lived. For the worker, it's actually even worse because it's one of the very first Darwinian paradox. Because Darwin himself, in his seminal book on the origin of species, wondered how sterility can be selected if workers cannot reproduce. Okay, it seems like a paradox that sterility can be transmitted by means of natural selection, and he qualified the case of ant workers as is one special difficulty, which at first appeared insuperable and actually fatal to his whole theory. And one of the solutions to this paradox, one of the first one has been given by Wheeler in 1911, where he proposed that a colony should be seen as a single entity and not a collection of individuals. It should be seen as a superorganism that is actually analogous to a multicellular organism, where the queen, so the reproductive caste, is analogous to the germ cells of a multicellular organism, while the workers that help the queen to transmit their DNA are analogous to the somatic cells that also help the germ cells to pass on the DNA to the next generation. And so this concept of superorganism has been seen as the endpoint of several major evolutionary transition in individuality. In this seminal book, The Major Transition in Evolution by John Maynard Smith and Eusociality, where the major transitions are the main ones are cellularity, multicellularity, and eusociality, and where the superorganism concept is seen as the most complex unit of selection because it encompasses all the other selection levels, because the superorganism is made up of multicellular organisms, which are made up of cells, which are made up of a collection of genes. And what is interesting is that from a developmental biology point of view, production of the different castes in the superorganism parallels the production of different cell types from embryonic stem cells, because both of these processes involve differential gene expression, meaning that a different set of genes are activated to differentiate into somatic cells or germ cells. And in the same way, a naive larva can differentiate into either worker or queen, depending on the set of genes that are activated. And what is really impressive is that in some ants, it appears that some of them exhibit remarkable ability to fine-tune the gene expression, because they have different castes, so worker and queens, but they also have different castes of workers with a large variety of size, shapes, and roles in the colony. And this is particularly obvious when you compare them to other ant species, where there is barely a difference among workers, but also between the queen caste and the worker caste. Here, for example, the queen is here, but can barely make the difference with the workers. So this means that we've got kind of another layer of complexity here, a complexity within the superorganism, where we go from simple eusociality with a very low caste polymorphism and a small colony size, and that go to complex eusociality with a high caste polymorphism and large colony size. And when I say large colony size, it can be really gigantic. So here you can see one of the most big superorganisms on Earth, a nest with this nest excavation of Atta levigata, where you can see how big they can be compared to humans. And so one of the questions that I have in my research is how eusocial genomes unlocked extreme phenotypic plasticity like that. How can we go from simple eusociality to complex eusociality? And for answering to this question, I will present you this paper published several years ago in Current Biology, where we sequenced the genome of 65 ant species. So with at least one representative from each of the 16 subfamilies. Basically, we also resolved the phylogeny of ants, where they are basically divided in three main groups. So the leptaniomorph, which are small species, pale, very subterranean and discrete species. some poneroids that represent approximately 10% of all ant species, and to finish, the big clade of formicoids that group all the major subfamilies of ants. Most of the ants that you know in the UK or in Europe in general are from formicoids because they constitute 90% of the ant diversity. And they nearly all emergences of complex Eusociality actually cluster in this formicoid clade. Okay, you can see it there, where the redder, the larger is the colony size, or the higher is the caste polymorphism. So you can clearly see that the complex Eusociality mainly emerge in formicoids and barely exist in poneroid and neptaniomorph. So the question that we have is, okay, from a genomic point of view, what explains the evolution of so many convergences to complex Eusociality in formicoids? And for that, we try to estimate the number of genes under positive selection on all the branches of the entry of life, with the question, are there some multiple positive selection events in formicoids corresponding to the multiple evolution of complex Eusociality? And when we do that, what we see is that we don't have multiple events of positive selection, but we have just one a major single positive selection hotspot here in the branch that lead to the formicoids, okay? So in this branch alone, so this is just a couple of million years, we have approximately 30 times more positive selection events than on the average of all the other branches, which is extremely unusual in this kind of analysis. And so one of the questions is, okay, what is the function of the genes under selection on this specific branch leading to formicoids? And what we see is that these genes are mainly genes controlling gene expression, such as functions such as like gene silencing by RNA, but the most dominant function is histone acetylation. Histone acetylation is well known for regulating DNA transcription, okay, so gene expression. And histone acetylation is also known to have some effects on key social functions, with some, for example, some experimental demonstration that when we inhibit the histone acetylation process in some ants, we can reprogram their caste behavior, but we can also enable worker reproduction or affect caste determination. So this suggests that there is probably a link with this hotspot of positive selection and the evolution of complex Eusociality. So it doesn't mean that the ancestor of all formicoids exhibited complex Eusociality, but it probably means that the molecular toolbox to finely tune gene expression to produce different castes from the same genome evolved at this moment. So we've seen that the key molecular adaptation for regulating gene expression may have evolved rapidly during a natural selection hotspot at the origin of formicoid ants, but it doesn't tell us what triggered the initial shift of gene expression toward either queen or workers. Basically, what determined caste, okay? And so what determined caste? For the male, this is simple. Actually, this is common to all Hymenoptera. When an egg is unfertilized, when the sperm doesn't fertilize the ova, the egg will spontaneously develop as a male, okay? So the males are haploid, and basically males doesn't have father, okay? They just have a mother. They just come from an unfertilized ova. For the females, so the queen and the worker, they come from a fertilized egg, and this fertilized egg can either develop as a queen or a worker. And what determines this shift is mainly believed to be environmental conditions. And for decades, caste has been believed to be uniquely determined by environmental conditions. I'm sure you all know the famous case of the royal jelly diet in honeybees, because in honeybees, when you feed larvae with royal jelly, which is a kind of high protein diet, the larvae develop as queens, and when you don't feed them with royal jelly, they develop as worker. And so because of this textbook example of honeybees, phenotypic plasticity has been seen as a central dogma of caste determination in social insects. But this central dogma has been challenged in the last decade with the discovery of several genetic factors that can affect caste determination. With actually some genetic lineage, for example, that tend to have more queens offspring compared to the average. There are some extreme cases of genetic caste determination that exist. For example, in Messor harvester hens with a complete loss of phenotypic plasticity, and where the caste is genetically entirely determined. And so this will do the transition to the next part. So the part that focus on the Messor genus, but we'll see how we can go from phenotypic plasticity to this genetically fixed caste. So first, a quick introduction about what are Messor harvester hens. So this is a genus of hens of around 135 species that are mainly distributed around the Mediterranean area, okay? And they are specialized in open areas and desertic, semi-desertic biomes, such as these landscapes. And they are very interesting because they are a specialist for consuming seeds, okay? They're granivores, so they spend most of their time to collect seeds. So you can clearly see them in south of France, for example, where they spend a lot of time collecting seeds. They're very ecologically dominant and visible, and they store these seeds in their nest. They're also characterized by the high caste polymorphisms, with in particular a caste of majors, which are big walkers with very big heads and massive muscles in their mandibles, with which they can crush the seeds, mix it to their saliva, and producing some kind of bread that they share with their nestmates and the queen. And so just I want to advertise a little bit a recent article about the taxonomy of Messor harvester ants, because we produced a data set and a phylogenetic analysis from 58 Messor species sequenced from museum specimen from the Natural History Museum of London, and that help us to redefine the taxonomic groups of the genus. I will not enter into taxonomic details. But we also performed some biological analysis that revealed that the genus emerged in the Irano-Indian area around 20 million years ago, before rapidly dispersing to the Middle East and Northern Africa. And this dispersal seems to be congruent with variation of temperature, because we can see higher speciation rates in during warmer periods. You can see here the temperature, when the temperature increases, the speciation rates increase, which suggests that warmer climates and aridification allowed Messor ants to expand and diversify in the Palearctic, and it highlights the strong effect of climate change on highly specialized taxa, such as Messor. Okay, so this is fascinating, but it's not what makes them so fascinating, because the most interesting things about Messor, the reason why they're my favorite ants, is because their caste is genetically determined. And we know that because of this species, Messor barbarus, a very common species in southern France, Spain, and northern Africa, because this Messor barbarus exhibit a very unusual reproductive system that is called social hybridogenesis. So this part is very important for understanding the rest of the talk, and is kind of complex, so that we try to go slowly to explain you what is social hybridogenesis, the weight reproductive system of Messor barbarus. So Messor barbarus is actually divided in two very divergent genetic lineage of queens, okay? So these queens are morphologically similar, but the genome is very divergent, nearly as divergent as two different species. And when we take them independently, this divergent genetic lineage of queens lost the ability to produce workers by themselves. That is to say that when a queen from the blue lineage mate with a male from the blue lineage, they can only produce new queens, they cannot produce workers. And it's the same thing for the red lineage. And this is a problem, because ants absolutely need to have workers to a functional colony. And the way they found to produce their workers is to kind of parasitize the sperm of the other lineage. So they engage in a kind of reciprocal sperm parasitism where they have to steal the sperm of a male of the other lineage to produce sterile hybrids that are used as workers. So in the end, all the workers, so 99% of the colony, are hybrids between these two genetic lineage. So, and also they can also produce males the normal way by laying some unfertilized eggs. So in the end, we have this very complex picture, which is called social hybridogenesis, where we got one species, okay, this is one species, Messor barbarus, but two different genomes that actually never mix except in the sterile caste, which is generation after generation, some first generation hybrids, actually they permanent first generation hybrids. And we got kind of four sexual types because whole the two blue males and the two type of males and the two type of females are necessary in a population to have a functional population. So it's kind of four sexual types. And the most bizarre thing about social hybridogenesis is that it is not an isolated oddity and that it seems to have evolved several times in evolution. It evolved several times independently in the Messor genus, okay, at least 3 times, but it also evolved in several other genera that are completely disconnected in the end phylogeny. And all these systems can vary. There's some variation of social hybridogenesis. The addition, for example, of queen or male clonality or other various differences, but all these systems have one common point. This is that this is a recurrent evolution of obligate hybridization for worker production. Okay, in all of the system, they have to produce hybrid workers. And so from what we discovered so far in all these papers, we can wonder, is it just the tip of the iceberg? Or do we actually underestimate the real prevalence of social hybridogenesis here? And to test that, we try to estimate this prevalence of hybridogenesis by developing a method to detect first generation hybrids from single genome data. And with this, we did a kind of phylogenetic scan from GenBank, and we found that approximately 3% of sequences of ants on GenBank are actually first generation hybrids. And when we compare that to other clades, for example, Hemiptera, Coleoptera, Diptera, or Arachnida, we found 0% of first generation hybrids, which indicates clearly that there is something specific about ants. So does it mean that 3% of ant species display social hybridogenesis or obligate hybridization for worker production? Well, not necessarily, because actually these hybrid workers that we find can be the result of facultative hybridization with retained phenotypic plasticity. Okay, so this is just the normal system, but just it happened that one queen mate with a male from another species and had some hybrid workers. But actually, this hybridization is for sure the first step towards social hybridogenesis, okay? And hybridization in general is typically counter-selected in most animals because of hybrid sterility. There is a famous case of the horse and the donkey. They form different species because when they mate, they can produce offspring, but this offspring, the mule, is sterile, which is an evolutionary dead end. And that's why we say that horse and donkey are different species. But in ants, hybrid sterility is not a problem. It's not a dead end, because in ants, workers are always sterile and they constitute a large majority of the colony. So we expect that hybridization should be less counter-selected in ants. And actually, hybridization should even be favored by queens, and it can be favored because we have sperm parasitism, where queens can promote hybridization with male from other species, because they may benefit from potential hybrid vigor in workers. So maybe having these hybrid workers is advantageous in the same way that mules are particularly robust and we produce them as humans. And so once we set this first step, hybridization and sperm parasitism, we just have a second step to have social hybridogenesis. We just need the loss of phenotypic plasticity. And this is actually expected to theoretically easily evolve in ants because larvae have kind of a selfish interest. Evolutionary speaking, they have a selfish interest to become queens and directly reproduce instead of act as helpers by becoming workers. Basically, they have an interest to avoid to be the altruistic caste. And you can easily imagine some kind of caste biasing allele that is able to bias the larval development toward queens. And such allele will act as a selfish gene that will propagate rapidly in the population by being overrepresented in the fertile caste, even if it's detrimental to the whole colony because it prevents workers to emerge. And so with this two-step, the fixation of the two steps should lead to obligate sperm parasitism, where the females need to hybridize to have their workers, and to test if this sperm parasitism and selfish caste bias can explain obligate sperm parasitism. We modelize an ant colony life cycle with various parameters that I will not enter into detail because I don't have time, but basically we made from this model some theoretical prediction and simulations. And what we found, so here I plotted some parameters of the model with the worker production rates. Okay, when it's 0, there is no production of workers. When it's 50, when it's 0.5, you got a 50-50 production of queens and workers. And here the sperm parasitism rate. And what we can see from here, the simulation stating values, is that first, you've got an increase of the selfish caste bias, okay? So it increased naturally because there is selfish interest for the larva. And in response, in a kind of evolutionary arms race, the queen are more engaged in sperm parasitism to counterbalance the overproduction of queens. And in a kind of a snowball effect, you've got a selfish caste bias that also increase. Then in response, the sperm parasitism that increase, and it goes toward this predicted ending point that correspond to the end of the simulations. That is to say, the worker loss, okay? The loss of the worker caste from intralineage mating. And what is interesting is that genetic caste determination evolves easily, even without hybrid vigor in workers, because we also test that with non-hybrid workers that will be less efficient than the normal workers. And we found that the worker production is lost even when the hybrid workers are twice less efficient than the normal ones, which basically means that an hybrid advantage is not necessary for the evolution of this reproductive system, and that simple conflicts among selection level are sufficient. So basically, you've got a selfish gene that enable a conflict between an individual of the colony, a larva, and the common good of the whole colony, the interest of the queen and the whole colony, which is to produce some workers. And so from this point, from obligate sperm parasitism, we can easily have evolution of reciprocal sperm parasitism, where just because the first lineage is parasitized, it starts to also parasitize the sperm of the other lineage, and you obtain the reproductive system of Messor barbarus, social hybridogenesis. And so we can wonder, okay, is reciprocal sperm parasitism the only possible evolutionary outcome in the Messor genus? And the answer is obviously not, because we recently found an alternative and even more bizarre route from obligate sperm parasitism, which is cross-species cloning that we explained in this recent paper called One Mother for Two Species via obligate cross-species cloning in ants, where we see how Messor queens became Xenoparity, meaning that they have to give birth to individuals of other species as part of their life cycle. So this introduces the third part about Messor from genetic caste determination to cross-species cloning. So this part started with a large-scale population genomic study on Central European Messor. And the first results that we have were this. Basically, from 300 genomes in 5 Messor species, we found that one of the species, called Messor ibericus, was engaged into obligate sperm parasitism by parasitizing the sperm of Messor structure. Okay, and Messor structure are just normal. Okay, they retain phenotypic capacity. They can produce both workers and queens. It's just Messor ibericus that need the sperm for producing the workers. Here are some data showing that. So you can see the 5 species, and you can see here the heterozygosity of the genome, which basically indicates if they're hybrid or not. And you can see that all the queens here, the triangles, have a very low heterozygosity. They're not hybrids, while all the workers, the circles, are hybrids. And we can see where come from the maternal genome and the paternal genome. And the maternal genome are all from Messor ibericus, while the paternal genomes are all from Messor structure. So this means that Messor ibericus strictly depends on Messor structure sperm to produce their workers, which implies that Messor ibericus distribution area should completely overlap the Messor structure distribution area. And the main problem that we had is that it's not the case. Okay, we got here, I plot on the map from our data set all the hybrid workers that we found, okay, all the colonies with hybrid workers that we found. And in red and blue, you can see the Messor structure distribution area and the Messor ibericus distribution area in blue. And you can see that in some places, both species overlap. So here, okay, it's logical. We understand how they can produce hybrid workers. But in some places, basically around the Mediterranean area, in Southern Europe, you can see that there is no Messor structure colonies that exist, which means that basically we can wonder, okay, how did the father come from? Okay, so basically how to produce hybrid workers without your partner species around. And so we search a lot for this paradox, because first we believe, okay, we probably have a big sampling problems. Probably Messor structure exists in Southern Europe, but we just don't find them. And we keep sequenced a lot, asked to myrmecologists if they already seen some Messor structure, and all of them told us, no, there are no Messor structure that exists, particularly in Italy and in Sicily, where we sampled a lot of individuals, and we didn't find any Messor structure. And the answer to this paradox is that actually Messor ibericus, as unimaginable as it seems, are able to directly produce Messor structure males. And when I say produce Messor structure males, I don't mean hybrids, but 100% Messor structure males with a full nuclear genome of Messor structure that is different from the queens of Messor ibericus. And for this, we got a lot of evidence. First, some field evidences where we found a lot of Messor structure males directly within Messor ibericus colonies, spread in 11 different populations. And what is interesting is that all these Messor structure males that we found in Messor ibericus colonies have Messor ibericus mitochondria that are identical to the queens of Messor ibericus, and actually identical to all the other nestmates of the colony. And as you know, so mitochondrial genome is a very small piece of genome, less than 0.1%, which is typically exclusively maternally inherited, which suggests that these Messor structure males have been directly led by the Messor ibericus queens. And what is interesting is that when you look at Messor structure males found in normal Messor structure colonies, they have the typical Messor structure mitochondria as expected, which means that this mismatch between the nuclear genome and the mitochondrial genome is exclusive to the Messor structure found in the Messor ibericus nest. We have, in addition, some lab evidence of the fact that Messor ibericus lay Messor structure males, because we kept and monitored Messor ibericus colonies in the lab, so something like 100 colonies, that we keep in boxes where they live in tubes, where we give them water and seeds. And what we did is that we genotyped the brood from 5 colonies for around 300 eggs and larvae. And when we genotyped them, we found that around 10% of these eggs only carry Messor structural nuclear DNA. And what we did to be 100% sure that these eggs were laid by queens, by the Mesorhybecus queens, we isolated Mesorhybecus queens in single tubes. And after 24 hours, we collected their first eggs. And again, we genotyped them. And we found that, again, 10% of these eggs only carry Messor structural nuclear DNA. We have also even more direct evidence of this phenomenon, where we directly observe in a colony from the egg to the emergence of the adults, a single queen that gives birth to the males of the two different species, which is a definitive proof that, as unimaginable as it seems, this is real. So this means basically that Mesorhybecus are able to perform cross-species cloning. So they can clone, basically they can clone Messor structural males from the sperm, which means that the nuclear DNA of Mesorhybecus ova is eliminated at some point before or after fecundation. We're not sure yet. And in the end, you just have Messor structural sperm that is in the ova that will develop in Messor structural male. So all in all, if we look more globally at how it works in Mesorhybecus colonies, so you have the typical normal mating between the Mesorhybecus males and females, where you got blue sperm, blue ova, and you've got just queens. When you got an unfertilized ova, you've got just Mesorhybecus males. When you have hybridization between the two species, you've got the workers, and sometimes you've got cross-species cloning, where there is elimination of the maternal DNA to give birth to an exact genetic copy of the father of Messor structural. So we're not sure, as I told you, about the exact mechanism. We don't know how the genome, the maternal genome is eliminated. It's probably analogous to other male clonality cases that have been observed in other ant species, in actually 4 different species. But this occurs at the intraspecific level, and here, obviously, the twist is that we've got Mesorhybecus that needs here to clone males from a different species in addition to its own males. And when I say that there are different species, I mean they really are very, very different species, because as you can see here, so from a single queen, you can have 2 males that belong to 2 different species that are not even sister species. Okay, you've got some other Messor species in between, and actually this is just the species from Europe. If you take Messor, the Messor genus as a whole, but even more species in between these 2 ones, and they are estimated to have a genetic divergence of approximately 5 million years, which is approximately the same thing than we have between human and chimp. And also from a morphological point of view, they're also very different, as you can see here. So the most obvious difference is the pilosity, okay, the number of hairs, because Mesorhybecus are very hairy, and Messor structural are really hairless, and hairs in ants are very important to distinguish species, particularly in males, because males of all ant species tend to be very similar. And here, this is really stunning to see such a big difference in terms of pilosity between 2 males that are led by the same mother. And what is wonderful is that these males are both useful to the mother, because these ones are necessary for producing queens. They produce sperm for producing queens, and this one produces sperm to produce workers. And so in the end, we have kind of a clear evolutionary scenario here, because we got an ancestral situation where we have Messor that parasitize the sperm from neighboring Messor structure colonies, okay? So in this situation, the two species overlap. And this go to a derived situation where Messor are able to lay and maintain a clonal lineage of Messor structure males for exploiting their sperm. And this is kind of similar, analogous to a domestication-like process, because Messor now favors and maintains reproduction of a species that it's used to parasitize from the wild, a little bit like humans maintain and favor the reproduction of domesticated species that it use to hunt from the wild. And this analogy with domestication seems even more relevant when you look at the internal phylogeny of Messor structure, because as you can see here, we have clearly two different lineages, a lineage that we call the domesticated lineage, which is only composed of clonal males, okay, that are found in Messor colonies, and also half of the genome of most of the workers that we found in Messor. And you have a way more diverse, wide lineage that regroup all the Messor structure that are found in their own nest, okay, in the normal nest of Messor structure, but also half of the genome of some Messor workers that prove that the ancestral system is still active in some places where the two species still overlap. And beautiful detail is that between these domesticated males and these wild males, we also have morphological differences, okay, with a typical clonal or domesticated morph, which are LS again, and wild type morph, which are hairy. This means that basically Messor ibericus interact with up to 3 males that are morphologically and genetically distinct. So basically, they can mate with up to these 3 males. So Messor ibericus, Messor structure, the domesticated lineage, and Messor structure, the wild lineage, and they can lay both of them, the Messor structure, domesticated lineage, and obviously they own Messor ibericus males. And so this introduced a novel reproductive system that we chose to name Xenoparity, from the Greek xeno, meaning foreign, different, and parity, meaning produce or give birth, which basically means that female are Xenoparous, which means that they require to give birth to the species individual as part of their life cycle. And this is kind of a big deal for this Messor ibericus, because by becoming Xenoparous, Messor ibericus queens allow themselves to expand their range by cloning these Messor structure males in their colonies and rapidly invading Southern Europe with their hybrid workers. Because to re-explain the evolutionary scenario that we have in mind, so basically we start with the initial situation where Messor ibericus needs to parasitize the sperm of neighboring Messor structure colonies, okay? So they can fill their spermatheca with the sperm of both species, so they can lay the queen caste and the worker caste. And at some point, they gain the ability to clone males of Messor structure by themselves. And so once they reach this Xenoparity, these Xenoparous Messor ibericus colonies became kind of self-sufficient, okay? And so they can now separate their range from Messor structure and invade Southern Europe. And so this means that we got here a kind of self-sufficient unit of selection, where the male and the female of different species intertwine completely their life cycle by becoming sexually dependent on the gamete of each other for their survival. And it kind of challenged our usual conception of what is a species, because usually when a male and a female depend on each other, sexually depend on each other for producing offspring, this is the basic definition of what is a species, okay? But here, clearly, this male and female that are sexually dependent are not from the same species. And this is clearly obvious from the morphology, the genetic, and also just the fact that the original male, actually the real male from the same species of the female, is still produced and still extremely useful because it directly produces sperm for producing the fertile caste. And so just a take-home message for finishing. So we've seen the superorganism concept that is kind of the endpoint of several major evolutionary transitions. And we've seen that transition towards complex eusociality may be related to a single short natural selection hotspot. We are also seeing that some simple evolutionary conflicts among multiple selection levels can lead to some very well reproductive systems, what caste is genetically determined via obligate sperm parasitism. And we've seen that from this obligate sperm parasitism, we can have some very bizarre superorganisms that are made up of two different species, where actually two genomes are part of a cohesive reproductive unit, which kind of mirror the two genomes of the eukaryote cells, where one species absorbs via endosymbiosis the genome of another species that became the mitochondria to give birth to a new unit of selection. And it is kind of the same thing in Messor, but at the superorganismal level, where the Messor colony kind of absorbs the Messor structural genome for having they worker and form a cohesive reproductive unit. And so this ends my talk. So with many acknowledgments of many co-authors, people that provided specimen, obviously thanks to my funding, so the ERC and for the Royal Mass Project, thanking also the Natural History Museum and my institutions. And also thank you for your attention, and a special thanks to Jay Hosler, who's a professor, which is also a cartoonist, and made for me this amazing cartoon that illustrates Xenoparity in Messor Ibericus. And so thanks for your attention.